This Week in Virology, the podcast about viruses, the kind that make you sick. From the 9th European Congress of Virology in Dubrovnik, this is TWiV, This Week in Virology, a special episode recorded on April 30th, 2025. I'm Vincent Racaniello. You're listening to the podcast all about viruses. Joining me today from Dubrovnik, Rich Condit.
Hi there, Vincent. Here we are again doing the Zoom live thing. This is cool. Dubrovnik is absolutely beautiful. The consensus amongst the conference participants is that every conference ought to be here. It is 70 degrees Fahrenheit, 21 degrees C today, clear blue sky. It's absolutely gorgeous. It's wonderful.
Well, I'm sorry I couldn't be there. I'm in the middle of my virology course, but I'm here in New Jersey where it is 19°C and cloudy. We have a couple of guests for you who are attending this meeting to give you a sense of the virology that's going on in Europe. Maybe that's the virology of the future, Rich. Who knows what's happening to virology in the U.S.?
Well, this meeting gives me confidence that virology is in good hands. We're okay.
Great. All right. Our guest today from the Plant Health Institute of Montpellier, Stéphane Blanc. Welcome to TWiV.
Thank you very much.
From the University of Rijeka, Croatia, Juranić Lisnić. Welcome.
Hi, I'm very happy to be here.
I left out your first name, Vanda. Sorry. I have my phonetic spelling pronunciation here, and I'm being very careful with pronunciation. And from the Medical University of Vienna, Elisabeth Puchhammer-Stöckl. Welcome.
Yeah, hello. Hello. Happy to be here.
So here's a couple of representatives of virology in Europe. Before we get into a little bit of science, Rich, what's this meeting about?
So, this is the 9th European Congress of Virology, okay, which is the virology meeting for the European Society for Virology, which I understand is something over 20 years old and meets every 3 years, but they're having such a good time that they're going to go to every 2 years. And I have to say, it's a privilege for me to be here. I have absolutely really enjoyed this meeting. It's been wonderful. There's great science, great people, great location, and I just love seeing the collaboration of all these people from all these countries coming together. And even look at the funding slides. The funding comes from all over the place. So it's a wonderful collaboration of people, and I've just really enjoyed being here.
So all viruses represented, Rich?
Yeah, as far as I can tell. I haven't seen anybody missing. Well, there aren't 16,000 presentations, but I don't see anything missing. And we have a good representation here. We're good.
Yeah, I'm not a fan of the 16,000 presentation meetings, Rich. It's too hard to make choices. Anyway, let's talk about our guests. We're going to first find out where they're from and how they got trained to become virologists. Let's start with you, Stéphane. Where are you from and how were you trained?
I'm from the south of France. I was born there and I was trained there, actually. Well, I did my training is very classical. I went to the university, I got a degree in Master on Evolution, then did a PhD on viruses. Then I started working on molecular interaction with viruses. Half of my PhD was in Norwich, in England, in UK. Then I came back to France and got a position at INRAE, which is the National Institute for Research in Agriculture and Environment. And then after I got this position, I went for a postdoc in Kentucky, actually. I was working on plant virus transmission for the first 15 to 20 years of my career.
Where in Kentucky?
In Lexington.
At the University of Kentucky?
Yes, in the plant pathology department.
So you've been into plants from the beginning, right?
Yes, I've been in the plants from the very beginning.
So
yes.
What is it about plants that grabs your attention?
Nothing, basically. No, I mean, I didn't know what I wanted to do. So I was sitting in the university, like many young people do, right? Then I find a training lab, hosting lab for a training period. And it happened that they were working on a plant virus.
Okay.
And so that's how it started. And then I realized I liked the lab. And because I think that a project, there's no bad project, right? From the beginning. If you ask someone to choose between saving the world from HIV or treating cauliflower, right? They will choose saving the world from HIV. I started trying to treat cauliflower. And then you build your own question, your own concepts, and then you tend to like it. And so I remained in this topic.
I don't know. I kind of like cauliflower. I might choose that too. So, Stéphane, why did you want to be a scientist in the first place? Was there some influence in your family or something else?
Again, same thing. My family they were selling hardware on the market, so there's nothing related to science. But they wanted me to go to the university, of course, not to do the same thing as they did. So we have no electricity anymore.
Okay.
And so I've been to the university because I was pushed by the family to do that, and I was good enough so I could, grade. But I didn't know what to do, and the first time I trained in the lab, then I realized that was cool, and then I decided to do research at this moment. But there was no special incitation or predisposition or whatever.
Well, the world is better off that you found yourself in a plant lab to start with. That's great.
So, Vanda, where are you from and how were you trained?
So I'm from Croatia, from Zagreb. I studied biotechnology and then also by pure chance ended up working in a virology lab. I actually wanted to do genetics, so I was a little bit disappointed to be working with mice and viruses, but now I'm actually quite happy to be working with viruses. I did part of my PhD also in the States on the Ohio State University. But I never moved from cytomegaloviruses because they are just the coolest viruses. They are huge. You can do whatever you want with them, and they can be both beneficial and bad.
So how did you first get interested in science?
I wanted to be a cell biologist since I was 10 years old. So when you are 10 and you start learning about cells and you see this picture of animal cell and plant cell, and when I saw this, I was like, wow, I want to be a cell biologist. And then, I ended up in biotechnology because I thought it's going to give me, if I don't end up in science, at least I can make my own beer and wine because this is traditional biotechnology. So.
And Elisabeth, tell us where you're from and how you were trained.
Well, I'm from Vienna. And I've studied in Vienna, and I'm a physician, actually. I studied medicine and actually wanted to be with patients and in the hospital, and this was my aim. And then more or less by accident, before starting my training, my clinical training, I came to the virology department to make some experiments and spend the time there for a project. And I found it so very interesting that I stayed, and I never regretted it. And as my father was a scientist as well on the veterinary side, it was not unusual to do science, actually, and not see patients.
Pretty common story that I've heard, right? Somebody trained as a physician and then just spends a little time in a laboratory and gets hooked. So have you ever practiced medicine, treated patients, etc.?
Only for a short time to make my practical part, but what is the different issue for me and my colleagues from the genetics, so that I have, as all my colleagues from coming from the medical part, seeing the patients always inside. So we won't go to basic science alone. We always have a patient in mind and what we can do for the clinic. So this stays.
And this is very important, I think, for the medical persons in our institute.
So in Vienna, are you in a medical school?
Yes, I'm in medical
So
school.
you're rubbing up against doctors all the time?
Yeah.
Okay, good.
So Elisabeth, did you want to be a physician first and then a scientist?
Yeah. But so it seems that you can be both in some way, because we are giving advice to physicians, and we now get questions on patients, of course, and we have a lot of diagnostics as well in the institute. And the translational part is, I think, the most exciting for me. Yeah.
And what caused you to want to be a physician in the first place? Was there some family influence or other?
No, I don't remember when, but it was long-term wish either to study medicine or music, and medicine was the more interesting one finally. And I'm happy that I did medicine and not music, but yeah, so it was actually my...
So do you play an instrument?
Yes.
What do you play?
Piano. And
okay.
So, but
cool.
Okay, let's move on to science. Let's start with Stéphane. Now, Stéphane, you work on nanoviruses and viruses as model systems. So can you tell us a little bit about these viruses, their structure, their genome, their host range, disease, transmission, the whole thing?
The nanoviruses first. They are single-stranded DNA virus and multipartite by essence. I mean, they are the viruses with the highest number of segments that are packaged individually. So the nanoviruses, in fact, there are two genera in this family, Nanoviridae. One genus is a nanovirus with 8 segments, and another genus is a babu virus for banana bunchy top virus. So they infect plant members of the family Bananaceae, I would say, which doesn't exist, but they infect banana and related plants, I would say.
I love it. Even a plant virologist giggles at the names of the viruses.
And the nanovirus on legumes, they create sort of problem for some crops, like in Sahel, in North Africa, on faba beans, on pea in Europe a little bit, but there are no big problem. In contrast, the banana virus is the problem number 1 in the world on banana crops. So it appeared in Far East Asia, and now it's invading the whole world. It's invading Africa, and it's reaching the States soon, I would say, right? And it's completely destroying the banana field. So that's a big problem. So that's for the nanovirus. And for the tospovirus, we started working on this because we thought that nobody cares about plant viruses, and if we wanted to have an impact on whatever we discover, we need to find a virus which may have some property in common with a multipartite virus, but that belong to a family or a group of viruses that also infect, mammals and eventually humans. And we have a Bunya virus in plant, which is called tomato spotted wilt virus. And so we start working on this because they package the 3 segments. The textbooks say that they package one copy of each of the segment, negative strand. But in fact, the packaging is a big mess. They package whatever. There's not a single particle that is identical to another one. And so in this virus, again, you find something reproducible in terms of composition of the genome only at the population level. So that's why we started working with this tospovirus to try to enlarge a little bit the impact of what we are doing on multipartite virus.
These viruses are all transmitted by insect vectors?
Yes, the nanovirus, most plant viruses, if not all, are transmitted by vectors just because the plant doesn't move. And also the plant is protected by a cell wall. So the virus cannot go through the cell wall. So they need a bridge to enter and to get out. And so the bridge is created by a third partner, which is the vector. And mostly arthropods, so it's mites, insects, it can be nematodes also. And so all plant viruses are transmitted by vector nearly. And for nanovirus, it's aphid, which is the most important vector for plant virus all over the world. And the tospovirus is transmitted by thrips, which are very small insects that
So you said, I just want to get it clear, you said that the packaging of the RNAs in the tospoviruses is a mess. So we have 3 segments, right? By a mess, do you mean some capsids have 1, some have 2, some have 3, could be anything?
Yes, by a mess, I mean that first the capsid size, the diameter ranged from 65 nanometers to 135. So you have a possible variation of volume inside the capsid of tenfold, right? And so they can package very variable amounts of RNA, sorry. And some of them package several copies of the same segment. Some are empty. Some package just 1 copy of 1 segment. And surprisingly, they are supposed to be negative strand viruses or ambisense, as we said. So they have a viral strand that is packaged and a complementary strand. And we found out that for some segments, actually what is packaged is the complementary strand in majority, like 60%. So this is what I mean by a mess, because we don't know what's in the particle in terms of the nature of the segment, the polarity of the segment, the number of copies of the segment. They are all different.
And the nanoviruses, do they package just 1 circle per particle?
Yes, this we are sure because we got the atomic structure of the virus. We can calculate the volume inside, and also in the whole family, right? They have either 6 segments or 8 segments. There are many species, but among the whole family, the size of the segment that is packaged vary from 985 to 1003. So there is a variation of 20 nucleotides. So there's no space for a second segment, for sure.
So that brings up the question I wanted to ask you during your talk, which is, the coding sequences for each of these segments must be different sizes.
Yes.
Right?
Yes.
And yet the genome, the circle in each case is the same size.
Yes.
So is there leftover DNA? Is there stuffer DNA? What's going on?
We don't really know. We know of some regulatory sequences for replication, for transcription, and for translation, but there are probably sequences we don't know, and regulatory sequence. Some of them must be included in the coding sequence, because, for example, the rep protein is the largest. It's 27 kilodalton. So this is occupying almost the whole space, while all the segments code for protein that are just 13 kilodalton. So there's a lot of space for regulatory sequence.
And that space, does it appear to be non-coding? I mean, the sequence.
I mean, the non-coding sequence?
Yeah, I mean, what does the sequence of that extra DNA look like?
Well, the origin of replication make a stem loop, that's for sure. And there's a second region that is highly conserved. That's for attachment of the master rep onto the DNA. And for the rest, there's no homology. So we don't know what it looks like.
All right.
I mean, the non-coding parts, right?
Okay. So how do you study these in the laboratory? Do you have plants in the laboratory, cell culture? Do you raise your own insects? How does that work?
Okay, cell culture, forget it. I mean, there are two cell culture in plants. Basically, it's BY2 cells, tobacco cells, and that they are not isolated cells, right? Some of them, they are making filaments, they are connected, and also Arabidopsis, which is the plant model plant. And some people have established cell culture, but they're making callus, which means they divide. So they recreate a cell wall, and they divide into callus. So it's like groups of cells. But I think the connection between cells doesn't look like the real connection in a plant, right? And it doesn't look like isolated cell either. So it's somehow useless in our case because they have a cell wall, so you cannot infect them. So we cannot use cell culture. So we have to infect plants. And because this virus is restricted to deep tissue, like the vascular system, you cannot rub the virus on the leaf. It's not going to infect, right? So we have to clone the virus in a plasmid, transform agrobacterium, that is a bacteria that is transferring DNA into the plant. And then we make 8 culture of bacteria, each with a clone of a segment. We mix the culture and then we infiltrate with a syringe the stem of the plant.
Okay.
And eventually we get an infection. So yeah, it's a bit complicated.
And insects, do you raise your own insects? How do you do that?
Yes, we have insectarium, basically. So we maintain colonies of insects.
Both aphids and thrips?
Well, for the thrips, we collaborate with the group of Anna Whitfield. They are in Raleigh, in North Carolina, and Dorit Rothenberg. And they are going to prepare the thrips for us. And we are transferring all the technology for RT-PCR, strand-specific, and things like that. But we don't do the thrips in France because the thrips is a real pain in a greenhouse. I mean, they invade the whole thing. So if we rear thrips in our plant health institute, it's going to be, people are going to be upset.
Okay.
So we rear aphids only, right?
Can you do experiments where you have an infected plant and then you let aphids feed on it and they will pick up virus and move it to another plant?
You mean experimental transmission?
Yes.
Yes, we do that every day. Yeah, that's very easy to do.
Okay, I have a question about how the genomes are replicated. So the nanoviruses are DNA. I presume they use the host DNA polymerase?
Yes, it does. I mean, the MREP protein is I was talking about the replication protein, not the replicase, because it's not a polymerase, clearly. So this protein is binding to the stem loop on the sequence of the segments, and then is having a helicase activity to open the DNA that has been made double-stranded within the cell. And then it's recruiting some host polymerases, probably polymerase alpha and delta.
It sounds like a T antigen-like protein, right?
Yeah. Oh, yeah. It does. Very much like.
Recruiting the polymerase to the origin of replication, right?
You were telling me you get sort of a rolling circle replication?
Yes, the replication is rolling circle replication, definitely.
So the TOSPO viruses must encode their own polymerase, right?
Yeah, the TOSPO virus is a negative strain virus. And TOSPO virus is really like Rift Valley fever virus or Crimea Congo or La Crosse virus or Ukuniemi virus. So it's pretty classical Bunya virus, I would say. Yeah. And works the same way. Actually, in the textbook, they are drawn and represented the exact same way with a polymerase associated to the RNA within the particle.
Now, I've read in one of your papers that the nanoviruses do not replicate in the aphid vector, right? So how do you maintain all of these segments in the population, or the circles in the population?
In the aphid, you mean?
Yes.
Well, many plant viruses do not replicate, are not supposed to replicate within the vector, even though they do a full cycle, right? So it's what we call circulative non-propagative transmission, while for classical arbovirus of animal, it would be a circulative propagative transmission, which exists also in plant, but it's not a majority. And so those viruses that don't replicate, they are supposed to accumulate massively during a few days in the gut cells, and then the gut cells slowly release virus, during the whole life of the insects. So once the insect has acquired the virus, even if it's removed from a virus source, it will be capable of transmitting the virus for, until it dies. So the reservoir within the insect, massive accumulation is supposed to be in the gut. And what is interesting in the case of the multipartite virus is that it's stored in vesicles, right? Which are supposed to release slowly the virus into the hemolymph. But this vesicle, we have shown that it is still importing new viruses. And I don't know of any vesicle in the cell that is doing that for several days, right? And that's the case here because we can see the acquisition of a secondary segment 3 days later, and then it is going to co-localize with the segment that has been acquired first. So this vesicle, they are storing, releasing slowly, but still importing new viruses from the gut.
But they are not replicating in the aphid, correct?
So far, they are not replicating. They are stored in a vesicle, and actually, when we do fish, we see this vesicle, they are all cytoplasmic, And this virus is supposed to replicate in the nucleus. And we'd never see viral DNA in the nucleus of aphid cells, while we always find the viral DNA in the nucleus of plant cells.
Okay, I have another 1,000 questions about that, but we'll just move on. You have an 8-segment virus in case of the nanoviruses. We did this this morning. How does the replication actually happen in the plants? Okay, do you get all 8 segments in a cell? Do you get how does the infection work in the plants?
Okay, so that's what one of the big discovery we've made is that the segment do not colocalize in individual cells in the plant. So, by the way, this is the sole so far biological thing that can replicate with a different gene in different cells, right? And so what we have partially demonstrated is that, for example, for the replication protein, it's produced within a cell where the segment encoding it is present. And then either the messenger RNA or the protein itself can exit the cell and colonize other cell. Because in a cell where a segment which is not encoding the replication protein is present and replicated, we don't find the segment encoding replication, but we find the protein encoded by this segment. So something is moving. We don't know at this point whether it's the messenger RNA or the protein, but we know that in plant, it's possible for both. The messenger RNA mobile in plant and protein mobile long distance in plant have been described. So we suppose that the virus is using those endogenous pathway to traffic the gene product and to make a sort of network of connection in between the different cells containing different segments.
So it's almost like the infectious unit is multicellular or something like that. You described it as complementation this morning, but it's and we're used to thinking of an infection happening on a single cell basis, but this is a collaboration amongst multiple cells with different segments in different cells, right?
Yes, clearly, and I think it's posing a number of questions. Like we haven't done single cell analysis yet, but this is just touching at the concept of differentiation for a virus, for example, because different cells have different segments, so they produce different proteins, and maybe the response of a cell is different depending on the segment that is replicating within this cell. So it would be nice in the future to do single cell analysis and to see what is the metabolism or the physiological condition of the different cell containing different segments. Because perhaps the response of the plant is differentiated depending on the segment that is contained. So that for the future.
So you, in one of your papers, you show that it's the virus particle that moves within the plant, not the genome. Is that something different from what you just talked about, the gene products or the mRNA moving within the plant?
The genome itself is moving within the plant as a package into virions. Mm-hmm. So the genome is colonizing newly formed tissue, right? And what we see is that we know we need assemble virions for the disease to go systemic. But when we look into the apex, into the young leaves, again we see the segments separated. So the virus is probably highly diluted in the sap, so it goes up in the young tissue, in the not fully differentiated tissue, and then is entering cell with a very low MOI, so that the segment doesn't invade together the same cell. They don't invade.
So one of the things you talked about this morning that was absolutely fascinating is the ratio of the segments in a population of virus, and how that's influenced by the plant, and I don't know if it's influenced by the vector as well. Could you just briefly review that for the folks out there in TWiV land and in the audience who may have missed your excellent talk?
Right. So basically, we were expecting to find equal frequency for each of the 8 segments in viral population, because obviously, if a segment gets really rare, it can be lost at transmission. So that was the expectation, because if there's no constraints on the frequency of the segment, the system should evolve to equal frequency to limit the risk of losing the rare segments, right? So we just experimentally assess this question, and to our surprise, we find out that some segments are 20 times more frequent than others, which means that for some segment, they represent only 2% of the total population of the viral DNA encapsulated. And so it means that those segments can be lost very easily. And it happened that those segments, the rare ones, they encode for the cold protein and for the replication protein.
You would think it would be the other way around, that those would be the precious segments.
Well, I don't know, because, for example, U4, I mentioned briefly this morning, one segment is called U4. U stands for unknown, so we don't know what it's doing. But what we know is that if we remove this segment from the infection goes perfectly systemic in the plant. The accumulation of the virus is the same. The virus is perfectly transmitted. So we don't know what this segment is doing. Maybe it's useful at some point of the ecological cycle, but we know that temporarily it's not useful at all. And this segment accumulates to a very high level. So maybe this is to avoid losing this segment when it is not useful, right? So I don't know whether we should expect what we should expect, actually.
Well, obviously we have too many expectations that this thing is defying.
Speaking of expectations, are there mammalian multipartite viruses?
Not known so far. So if there are some, they must be very rare or completely hidden or asymptomatic. I don't know, but I don't think they are. No one can say that someday we will find one, right? But it's not only mammalian, it's vertebrates in general, right? The only multipartite viruses that are surely infecting animals are the multipartite virus of plants that are transmitted by insects where they do replicate, right? Some of them are classical arboviruses and they replicate in insects. So for example, there are some rhabdovirus with 2 segments. The Bunia viruses, some of them make only ribonucleoprotein, no particle, right? So, and they have 3 or 4 segments, and so they are 3 or tetrapartite, and they replicate in the insects. So those, we are sure they do exist in animal, but that's it, basically.
So is there something special about plants that support a multipartite virus replication that animals can't do?
At least there is something in plant that is much more developed or perhaps easier than in animals is the trafficking of different gene product in between cells. I mean, because in plants you have a symplastic continuum through the plasmodesmata, and so traffic of RNA or proteins is perhaps much easier than in animals. Although there are cell connections in animals, and we know that some virus, like influenza virus, for example, is increasing the nanotube connection in between cells. And we also know, I mean, it's been published by the group of Anis Lowen on influenza A virus, that through this connection, influenza virus can exchange segment. But they haven't tested so far whether they can exchange RNA or proteins. So maybe this phenomenon is also existing in between animal cells infected by viruses, but it's not as evident as in plant.
So next time you're enjoying a banana, think about this guy. And I want to be reincarnated as a plant virologist, I think.
Okay, we'll remember that, Rich. All right, let's move on to Vanda. So I want to talk about cytomegalovirus with you, because that's one of your interests. So tell us a little bit about CMV, transmission disease, and so forth.
Okay, so cytomegalovirus is a herpes virus. Herpes viruses are very, very successful viruses. As far as I know, they infect every vertebrate and then even some invertebrates. And they are also very well distributed among the population. Again, everyone has at least one. I'm actually CMV negative and proud of it, despite working with it for such a long time. But I guess this is just pure chance. I got EBV, so that's fine too. And cytomegalovirus is, like all herpes viruses, large double-stranded DNA virus. It has tons of genes. We are still not entirely sure how many genes it has because we keep finding new ones, small ones, big ones, new transcripts, everything. And again, like all herpes viruses, once you get it, you are infected for life with occasional reactivation. So it waits and bids its time until your immune system is not up to scratch, and whoops, there it is again. And people who have herpes simplex know this the best because they usually always get it on the same spot.
Is there something special about individuals like yourself who are never infected with a herpes virus?
I don't know. I think maybe it's just pure luck, and the virus is just bidding its time to infect me, I'm sure.
So what fraction of the population is infected with CMV?
Okay. So this depends on the socioeconomic status. In less developed countries, 100% of people have CMV. In more developed, 50% approximately. Okay. And it's a trade-off, Sometimes it's actually better to be infected, especially if you're a woman planning pregnancy, because you will have your immune system and you will have antibodies generated, and this will prevent the transmission via placenta. Whereas if you get infected for the first time during pregnancy, then there is some chance that the virus will pass the placenta, infect the fetus, go into the brain, and then do damage.
So, my understanding is that one of the most serious disease outcomes is when a fetus is infected, mm-hmm. or a neonate as well.
Yes.
So I don't understand the circumstances of that. From what you just said, it sounds like you can be CMV positive, but if you're not actively replicating CMV, you're okay, with your kids okay? So under what circumstances can the fetus or the neonate be infected and suffer these consequences?
So I don't think this has been completely answered yet. It's not really my area of expertise completely. It's certainly known that if you are infected before and you have generated antibodies against the virus, they can be protective. Also, other immune cells, of course, are protective. But sometimes antibodies can be actually detrimental because they can help the virus to pass the placenta. As far as I know, the worst outcome is if you have acute first-time infection during pregnancy, because then you're still mounting your immune response, especially adaptive immune response, and then in this case, that's the worst outcome.
In that case, Vanda, is there any treatment that's done, or is it nothing to do?
So despite decades of research, we still don't have a working vaccine, although a lot of people have tried. You can use hyperimmune serum that sometimes help to block the virus with antibodies. Now we have some drugs which have a bit less toxicity than they used to before, but I'm not sure they've tried them in pregnant ladies yet.
Is there, under normal circumstances, is there any effort made during pregnancy to make an assessment as to whether or not the mother or the fetus is at risk?
Yes. So cytomegalovirus is considered a torch pathogen, so a pathogen which is quite dangerous for pregnancy, and women are routinely serologically tested whether they have antibodies. Go ahead. But despite all this, a lot of people have never heard of cytomegalovirus. So if you ask any pregnant lady who is not a virologist, she will know about Down syndrome, she will know about toxoplasma, but she will not know about cytomegalovirus. There is significant outreach, especially in the States. There is this very nice patient organization called Stop CMV, who is really trying to get the message across, because CMV is heavily disseminated among small children. It's secreted in the saliva. So, in a kindergarten, you have breeding grounds for the virus. So if a woman is pregnant and has never been infected and has a small child, this small child can be sort of dangerous to her. So people should know about this to take some precautions, maybe wash their hands a little bit more or
So under what circumstances is there a disease manifestation, and in what age groups and what sort of diseases?
So in general, as long as your immune system is more or less okay, you will have no symptoms, or maybe you will just have a few days of flu-like symptoms and you would forget about it. But despite that, you can secrete the virus in your saliva and in your milk for months. Actually, pregnant ladies who are latently infected with CMV very often will reactivate the virus and secrete them in milk and infect the babies like this. But if you are immunologically suppressed, then because this virus has a very wide tissue tropism, basically, I don't even know if there is a tissue that cannot support infection. The majority of organs we looked at are quite well infected. Therefore, you can get all kinds of diseases, retinitis, lung damage, inflammation of the liver, everything.
And neurological diseases in children, right?
In children, yes.
Okay. So if it infects all sorts of tissues, is latency established in a wide variety of tissues as well?
Again, excellent question. For human cytomegalovirus, monocytes and macrophages and hematopoietic stem cells are considered to be major source of latent virus, but it's a bit difficult to test anything else. Since I work with mouse cytomegalovirus, which is quite related, and then you can do stuff with mice which you can't do with humans, we very often check if we detect a virus in certain organs. We very often check whether they can reactivate from latency there. And so far, in many organs that we checked, you can definitely detect the virus.
So is there can you tell me how reactivation occurs, or is there a little bit of virus happening all the time? What's the story?
Yeah, again, very good question. Not entirely solved. So some herpes viruses have latency-associated transcripts, and I'm sure Elisabeth will talk a little bit about this, but not cytomegalovirus as far as we know. There have been efforts using next-generation sequencing. There is an excellent group from Israel that does it, and they never found any latency-associated transcripts. So what we think is that it's just a virus peeks its head above the ramparts, looks around whether there's an immune system that slaps him down. And this is why cytomegalovirus is, again, to my knowledge, the only virus which causes this immune inflation. So usually when you get infected by a virus, you get your immune response, your T and B cells, and they proliferate, they deal with the virus, and then they contract, and that's about it. You either have constant level of virus-specific cells or they go down. But for cytomegalovirus, after a while, the amount of your virus-specific T cells increases. And this might be due to this effect of virus occasionally reactivating in the tissue and then boosting and priming CD8 T cells.
So you mentioned mouse cytomegalovirus as a model system, and I've seen this used in your talks, and I'm always wondering how good a model is that? In what ways does it accurately reflect human CMV, and in what ways does it differ?
So in many ways, it reflects very accurately. These viruses are highly related. There are numerous genes which are homologous or have homologous functions. Actually, a lot of the translational stuff happened based on research done in the mouse and then translating this into humans. Immune response is very, very similar. We also, in mouse cytomegalovirus, we also see this inflation. Another characteristic of cytomegalovirus is a natural killer cell memory. So natural killer cells are innate immune cells. They shouldn't have memory. They should be jacks of all trades and not recognize specifically any viral antigens, just some pattern recognition, but not so for cytomegalovirus. So there are NK cells both in humans and both in mice which have receptors which have actually evolved to specifically recognize antigens from a cytomegalovirus. So these are similarities. So I've also told you that congenital CMV infection is one of the most worrying aspects of this virus. And here is where the model is not so perfect, because mouse and human placenta are built a little bit differently. Please don't ask me details about how the placentas are built, because I don't entirely know. But mouse cytomegalovirus cannot pass the placenta, whereas human has. Human can. But we are still dealing with this. In a way, baby mice, when they are born, they are equivalent of a third trimester babies neurologically, because they are completely blind and naked, and So we can actually infect the mice on the day that they are born, and then they practically have all the hallmarks of children being congenitally infected. So we see that their cerebellum is smaller, and you can also see this in humans. You see inflammation in the brain, you see recruitment of T cells. I mean, my colleague Ilya had really a nice talk about it, and that's actually something that he does.
Where did mouse cytomegalovirus come from originally? How long has it been around? I mean, how long have we known about it?
As far as I know, it has been co-evolving with the mice from some common ancestors that we have with mammals. So all of our viruses are very highly adapted to us. They have been co-evolving for us for billions of years. So I don't know exactly.
So how was it discovered? Do you know?
Yeah, it was discovered by a woman, actually, Margaret Smith, which is why the first strain of cytomegalovirus, which is actually one of the most commonly used still, is called the Smith strain. Okay. That I don't know any funny story about how it was discovered.
Does MCMV establish latency in the mouse model?
Yes, absolutely.
And how do you reactivate it?
Okay. So this is one of the discoveries that my original boss, Stéphane Juranić, made, is that the immune system uses multiple layers of protecting from reactivation. So you have to kill the immune system, and you have to kill it almost completely. You have to deplete CD4, CD8, and B cells, or you just do sublethal radiation, and then it is going to reactivate.
So let's talk a little bit more about NK cells. You mentioned they're really important for defense against CMV. I presume that's also true in the mouse model, right?
Yes. So yeah, natural killer cells are innate immune cells which should react first. And both in mouse and in human, I almost said in human model, both in mouse model and in humans, NK cells recognize the virus very quickly. Actually, people which don't have functional NK cells are quite susceptible to CMV. Now, there are some differences between humans and also between different mouse models. So the most commonly used mouse strain, black 6 mice, so little black mice, they have this one peculiar receptor, which a lot of people think has been developed as a mouse response to mouse cytomegalovirus. And this receptor is called LY49H, and it recognizes a viral protein. So one of the things this virus does is very efficient immunoevasion. It evades every aspect of our immune response. And because T cells are very good killers, what this virus does, it uses several proteins, depending on whether I'm talking about human or mouse cytomegalovirus. It uses several proteins to remove MHC1 from the cell surface. If there is no MHCs, then T cells are blind. But on the other hand, you have NK cells. And NK cells are monitoring your cells, whether they have sufficient levels of MHC, because MHC downregulation is a hallmark of a virus infection and also of a tumor transformation. So these NK cells have inhibitory receptors which recognize your own MHC, and this prevents them from killing you. And this works very well, which is why you don't have any autoimmunity based on NK cells. So cytomegalovirus developed additional proteins which will return some MHC to the cell surface in order to engage inhibitory receptors. And then the virus responded to this by developing MHC decoys. So molecules that look like MHC, but they're not presenting any peptides, or they are even offering fake peptides to MHC molecules, again, to fool NK cells. That's what human cytomegalovirus does. And then mouse and humans responded by developing activating NK cell receptors, which recognize these either changed MHC molecules, the ones in the complex with MHC, we are calling them altered self-MHC molecules, or proteins which look like MHC molecules.
Wow. So complicated. It sounds like virtually every aspect of the immune response is engaged in keeping this under control, and there's a constant tug of war going on with every aspect.
Yeah, absolutely. And this is why cytomegalovirus is very popular among immunologists as a tool to study, because if you discover that CMV is targeting some unknown molecule, you can bet it's going to be very important, especially in the immune response.
So I heard a rumor, I don't know if this is fake news or whatever, that some ungodly fraction of my antibody response at my advanced age is directed towards CMV if I'm CMV positive. Is that true?
And also ungodly amount of your T cell response.
Okay. So
that was Felicia Goodrum. She was on TWiV and she said that, remember?
Okay. All right. So that's not fake news. Good.
No, it's not fake. But this is a kind of self-serving question, Vanda. Do you know, have you heard of CD155?
Sure. I guess.
So CD155 is an NK cell recognition molecule, right?
Yeah, there's tons of them, so it's hard to keep track of all of them.
Well, I mentioned CD155 because I discovered it as the poliovirus receptor many years ago. That's why I say it's self-serving.
No. I'm glad that you mentioned it, because we actually work quite a lot on the poliovirus receptor in the context of CMV, not myself, colleagues of mine. Yeah. And we have also developed some antibodies against it, which are now being tested as a checkpoint therapy, but that's not what I do. And it's not really that connected with viruses, but yeah, it's a very, very useful molecule.
One of your other interest is CMV replication in ovaries, right? Can you tell us about that?
About CMV replication in ovaries. Yeah, that's a project I sort of stumbled upon. Like most of my scientific research is something I accidentally discovered. So this actually research started even while I was in high school. I wasn't working on it at that time. So Stéphane and his team, they wanted to see if they can force CMV to pass the placenta somehow, and they used a LAC-Z expressing virus, and they've never managed to force the virus to pass the placenta. But while they were doing this, because viral infected tissues become blue, they observed that ovaries were really, really, really blue. And then they were sort of sitting on this for a while, and then started digging when I came and some other people, and we discovered that cytomegalovirus is actually infecting the ovaries really, really well.
This is in mice, right?
This is in mice, but there are reports of viral DNA being found in human ovaries too.
Okay.
So we looked at the acute infection, and then we noticed that you would see, in many tissues which we consider highly infected, you would see a few infected cells and then nothing, and then a few infected cells. And in ovaries and in adrenal glands also, we see huge areas which are totally infected. And now in ovaries, you have two important tissues. One important tissue is ovarian follicle, which is a thick layer of cells surrounding the oocyte, so it's protecting the oocyte. And when a follicle ovulates, erupts, the remaining cells that stayed in the ovary, they differentiate and then they form corpus luteum. And corpus luteum secretes progesterone. And progesterone maintains pregnancy. And the virus has really high tropism and really high replication inside the corpus luteum, and we've never seen infection in the ovarian follicle. And then we did some research as to why that is, because these cells in the corpus luteum and in the follicle, they're practically highly related cells, because corpus luteum differentiates from the follicle, so you can infect them ex vivo. If you take these cells out, they can be equally well infected. But your immune system, and especially your innate immune system, is there to prevent this from happening. And it makes sense, because since we've been co-evolving with the herpes viruses, if herpes viruses would by any chance go into the follicle and kill your oocytes, then we would not be here. So viruses, if you, for instance, put a vaccinia virus, which is not a natural mouse pathogen, into a mouse, it sterilizes it. So it's, I guess, expected from a natural pathogen to sort of, a natural pathogen which is causes chronic latent infections to sort of be at peace with the host.
So is there any evidence that human CMV can have any impact on fertility or anything like that?
Not really, I think, but I think that's really hard to research, because how are you going to get ovarian tissue? I was thinking about it intensively. You can only get if there is a suspicion on ovarian cancer, but then if the cancer is there, a CMV sort of likes to go into cancer, so this could give you false information. But there are now some clinical studies which are trying to connect CMV with the ovarian cancer. And that would also sort of imply that maybe it's there reactivating, changing a little bit of what's happening in the cell and maybe causing cancer.
Okay, thank you, Vanda. Let's move on to Elisabeth. Elisabeth, I want to talk about Epstein-Barr virus and MS. Okay, I know you work on a lot of herpes viruses, but I want to talk about that one. So tell us a little bit about EBV, transmission, diseases, and so forth.
Yeah, EBV is probably the herpes virus. Everyone is infected worldwide. Although most people don't realize that they are infected. It's the same for many herpes viruses. And you get infected with this virus either in childhood or in adolescence or at young adult age. But when you are 40, most people are infected with EBV. And it's seen, we have this Klimt picture in Vienna, the kiss, and we always show the kiss as a close contact between persons, so transmitting Epstein-Barr virus. So if you come to Vienna, you see the picture for Epstein-Barr virus we are all using all the time. And it needs a close contact, but it manages to come over to nearly everyone over a lifetime. And many people don't have any problems with EBV over their lifetimes, and others have enormous problems. It starts with the primary infection. Some people have infectious mononucleosis, which is a clear picture of a sore throat and lymph nodes swollen and lung implications. You can have an exanthema and involvement of the liver and so on, high fever. And it is actually, for me, the first virus where I was associated with a long-term fatigue, which is now a big topic after Corona, of course. And some people don't have any symptoms. And the question is always, why do some people get symptoms and others don't? And so I've presented our data that it's a human genetic factor which makes the difference. And if you have a certain genetic variant, you are very unlikely to have infectious mononucleosis if you have this homozygous form.
I wanted to know what the site of latency was and the mechanism of reactivation.
Yeah, there are probably a number of reasons why you may reactivate EBV. Mostly you don't realize it. But now, as we have these people with long COVID or whatever, people tend to make some, if they are tired or so on, they come to the physician and make some tests and look for PCR or look for antibodies. And it's something we don't like, actually, if people come and I'm so tired and so, and my physician says, I have antibodies against EBV, so I'm sick, and this is my reason to be sick. And we all say, we also have antibodies against EBV. So it's a misunderstanding, I think, in the population that simply because you have EBV, it's not necessarily the reason why you are tired or have some infection or some disease, diffuse disease, where you don't find any other reasons. But everyone is from time to time reactivating. We have made some studies in the healthy elderly population, and you find in about 10 to 20%, you find EBV DNA in the blood. So there is constant way of reactivation, and probably also in the throat. Yeah.
So we'll get to genetic factors controlling disease and latency, but I have this thing in my head that that mononucleosis is associated with a later exposure to EBV. Is that true?
Not really. We also thought this, and this is the reason why we specifically looked on the genetics also of the children's population. And it seems that the same factors bringing children to infectious mononucleosis as adults. But maybe from some reason it's a bit more mild, but they also have infectious mononucleosis, and they also may get over early to multiple sclerosis in an earlier age. So this is possible as well.
Okay, so I got to change that one.
Yeah, it was also for us, the idea that children are different, but not so much different.
Okay.
So can you tell us exactly what is multiple sclerosis?
Mm-hmm. Well, multiple sclerosis is probably autoimmune disease, which starts quite early in overall in the age between 20 and 40 years of age. Maybe a bit earlier, a bit later, but not much later usually. And it starts actually with the clinical symptoms. You diagnose this when the patient comes with clinical symptoms. They may have some unexpected blindness or a problem with walking or with other impairments, which may then release again. And usually then the clinical diagnosis is made. And it's some nodules or lesions in the brain, and this can go back, but get more and more in many patients. Not all. Some have a constant one episode and then it's over, but many people have relapsing, remitting procedure and disease, and it's not, you cannot really heal it, but there are a number of drugs you can give to try to limitate the impairments of these persons. And the problem is that it starts, or one of the problems, that it starts at young age and you have a long life with these impairments then, and therefore it's in a strong interest on this disease. Yeah.
So the actual pathology, what at a cellular level, what's going on?
It's the glial cells and the neurons is distracted. The glial surrounding is distracted, and is sort of autoimmune on different levels in the brain. And the autoimmune antibodies, as we have seen and others have shown within the last years, are against a number of different proteins. It's the myelin basic protein. It's a glial CAM, which is an adhesion molecule leading to efficient adhesion of glial cells. So on a variety of cross-reactive proteins or similar proteins, that the cross-reactive antibodies directed. And Epstein-Barr virus was seen since really many years as a potential cofactor or factor triggering multiple sclerosis. And we have always seen there is something. And there was this epidemiological study in 2022 in the United States. You know them. It's probably all where they looked at millions of people and said there is epidemiologically, in fact, a risk for multiple sclerosis if you have had infectious mononucleosis. And then it needed to be really defined where is really the point between EBV and multiple sclerosis.
So what were the first hints that EBV might be associated with multiple sclerosis?
Well, it was said, this is what I learned already, or I was teaching our students, that there is, if you have infectious mononucleosis, you have an increased likelihood to get multiple sclerosis within a few years later. This was said, but it was not so really confirmable. And the second point is that if you are infected with Epstein-Barr virus, you have a number of antibodies, and a part of the antibodies are directed against the EBNA1. And some people don't have EBNA1 antibodies, but most have. And if you have multiple sclerosis, it was always shown that there is somewhat higher antibodies against EBNA1. But it was diffuse, and there was a significance, but you could not really figure it out, and it was very diffuse.
So the 1922 epidemiological study, that was the one in the military, is that right?
2022. Yeah.
Is that's very significant? Is that correct for firming up this association?
Yeah, it was very helpful.
Okay.
And it showed it in a big, and it was an interesting thing because they showed also a minor association, protective factor for cytomegalovirus. That cytomegalovirus is a little bit protective, but it was, and we could show that this in fact is the case, but you need to have a specific cytomegalovirus strain in the UL40 in a certain region, and you must have a human genetic profile which fits exactly to this strain. And if you have both, then it's protective, and if not, it's independent. Doesn't matter at all what you have. So you always have to look in detail, personalized. Yeah.
So, Rich, you said 1922. That's a 2022 study.
Yeah, well, I'm an old guy. I don't. Okay.
Yeah, I got it. We did that paper on TWiV.
Yeah, that I remember.
Okay. So, Elisabeth, one of the treatments for MS is giving a monoclonal against B cells, right?
Yeah, there are a number. I've taken time together with our neurologists to go through all the therapeutic options, and there are lists of therapeutic options and coming more and more. You usually start with immunosuppression, with slight immunosuppression, and then go over to monoclonals. But if you really start with the monoclonal antibodies, some can trigger JC, as you probably know. Mm-hmm. So it's not undangerous, and better you can start early with a slighter scheme and not, yeah.
So we now know that the problem is that these antibodies to EBNA cross-react with a neuronal protein, right? Glial, CAM. How do those cross-reactive antibodies cause MS? Do we know?
Well, it's antibodies against human proteins, which directly may act exactly. I cannot figure out how exactly these antibodies work directly in the brain, but we suppose they're just there and make some destruction, as usually autoantibodies do, and not different to other schemes. But it's also interesting maybe that also healthy persons may have some level of autoantibodies, but from some reasons they have mechanisms to get rid of this much better. And we have looked on a test system to figure out who has really the malignant way of autoantibodies or cross-reactive antibodies. And we needed some tests over time to not just one single time point after infectious mononucleosis, but you have the infectious mononucleosis, and then 9 months afterwards we would start to look at these antibodies. And then within 3 years or 2 years, we have a few samples to test. And if you're constantly high with these autoantibodies, you 100% likelihood that you get multiple sclerosis, and that it will be quite rapid if you're always positive with these autoantibodies. So this is, yeah.
So what fraction of individuals that are infected with EBV develop autoantibodies? Is this a
develop multiple sclerosis?
Autoantibodies.
Yeah, this is not clear. That it's also not clear. There are some persons we know since quite a long time, all the diagnostic people in this room, that about 10% of the persons don't develop EBNA antibodies at all. So we have a problem in the diagnostic, makes a problem because they're unusual, so they're not having antibodies. And from the others, some people develop autoantibodies, but maybe for a short time and then get rid of it to some way. And there, for instance, the NKG2C immune response is an important one, triggered by cytomegalovirus, which puts away the producing cells, and so very complex. Yeah.
Does the age at which you're infected with EBV make a difference for the development of autoantibodies?
I don't know yet. We have looked on more or less adult persons. There we cannot find a difference. Maybe it's more female. But with the children, there are now studies going on children also with some US cohorts currently to look at these antibodies and with these tests to see whether we can also predict in children, and it's about the same antibodies as in the adult population. So there seems not be.
So maybe you could briefly go over what you described in your talk about the genetic factors from both the virus and the humans that impact whether or not your EBV infection is going to lead to multiple sclerosis.
Yeah, there are a number of factors, but the two main factors, when they come together and have the most significant prediction for multiple sclerosis, is the one is a genetic factor of HLA-E. This is a non-classical MHC molecule, and there are two alleles, two variants, different in one amino acid actually, the 1,1 and the 1,3, and this is the one side. And if you have the 3 variant, it's more protective, and the 1 variant is less efficient, and therefore you have probably more virus and less good prognosis. This is the human side. And it's bad if this comes together with a virus strain with a certain variant in the LMP1, because there are some peptides presented by this HLA-E, and these are limited. You cannot have hundreds of peptides presented. You have, for EBV, we have found 2, and now we are possibly 4 peptides of the whole virus, which may be presented by this way. And one of these is LMP1. In LMP1, a variable region, and there are 11 different variants, at least in the Austrian Central European population. And if you have 1 or 2 of these variants, and they come together with this HLA-E1 homozygous form, then you have a 260-fold increased risk to develop multiple sclerosis.
And is that because, how's that related to the autoantibodies?
It is related to the autoantibodies, yes, because you have more virus, you cannot get rid of this virus. This is the hypothesis, that you cannot get rid of this virus so efficiently, and therefore you produce more antibodies.
Okay. So what's the prevalence of that particular HLA type in the population?
There are 2 variants, and actually it's equally or similarly distributed. We have the heterozygous form is about half of the persons, and then you have 20-something 3 and 30 nearly 1. So this is it's the same for the infectious mononucleosis. If you have this 3, you're unlikely to get infectious mononucleosis. It's somehow
Is EBV the only cause of MS, or there are others?
We have, from our data now, it seems that we have 4% from our cohort where there is nothing, it has nothing to do with EBV. They have neither autoantibodies, nothing. And this fits to what our neurologists say, that there is a minor subgroup or where there is another reason. And we have, of course, this has to be investigated. Yeah.
So what are some ways that MS could be prevented, at least the EBV-caused MS?
Well, the concept is actually that from the actual data, I mean, it has to be worked out still, that if you have infectious mononucleosis or your child has infectious mononucleosis, you wait 9 months and then you test. Currently, this would be my concept, within the next 2 to 3 years, 3 or 4 times for these autoantibodies. If you have constantly high autoantibodies, you have a high risk, and then you immediately go for early radiography, for instance, because the brain lesions are earlier than the clinical symptoms. And when you get with therapy in the situation where you have the brain lesions starting, you can probably postpone or hopefully postpone the clinical symptoms for a much later time point. And this is actually the concept our neurologists try to get over now. I mean, the question is whether how long it takes to convince people to test their children when they have infectious mononucleosis, but this is another question.
Is any of the pathology reversible? So can you anticipate treatments that might help a person who has symptoms recover? The symptoms can recover when you have treatment, not in all persons, and it's not forever, probably, but you can decrease the symptoms and maybe limit the development of the lesions, but you cannot get totally rid of the lesions if they are there. In my understanding, I'm not a neurologist, they are there. But they're growing, can be, or the extent can be limited by or immunosuppressive treatment.
Do you think that a EBV vaccine is feasible?
Yeah, there are some companies working with EBV vaccines, very unsuccessful so far. And it's also the question whether antibodies will help. And after my experience or our experience, you must be extremely careful what you use as a vaccine, where you get antibodies against. I hope everyone is aware of the autoimmune problem of EBV. And there may be some possibility to have a vaccine with short peptides against, also conserved peptides in EBV. We're also working on this in a bit, but currently there, I don't see
It's interesting with a virus that is that prevalent. Okay, yeah. I have to wonder what the impact of eliminating it or altering its biology significantly would be on the population. You don't know. Very interesting.
Yeah, but for the vaccine, if you are transplanted and you know you have a transplantation as a child, for instance, then you should have a vaccine before transplantation. This would be the first, I think, option.
Same for HCMV, right? And are there vaccine potentials for HCMV?
I mean, vaccines have been tried to be developed for many years. There are now some mRNA vaccines in the works. We'll see, but
vaccines against herpes viruses seem to be very problematic.
Yeah.
Except for shingles, right?
Yeah.
Yeah.
Shingrix is great.
Yeah.
Shingrix is great.
Yes,
right.
That's true.
Yeah.
All right. I have one We have one more question for all of you, and then we'll wrap this up. Let's start with you, Vanda. What would you have been if you had not been a scientist?
I don't know, biologist, I guess. Maybe I would grow some plants. I grow some plants as my side job, and now, not a side job, as my hobby. And now that I've learned a little bit about plant viruses, I will not feel so bad when I kill my house plants. I will just blame it on the virus.
Stéphane, what about you? What would you have been if not a virologist?
A fisherman, maybe.
There you go.
Somewhere close to the sea, yeah.
You like to fish?
Warm sea. I like fishing, yes.
But Montpellier is not close to the sea, right?
It's 8 kilometers.
8 kilometers. Oh, that's not bad. Okay.
Not bad. Yeah.
I don't know my French geography, unfortunately. Probably you don't either, Rich.
No, but I knew where Montpellier was.
Elisabeth, well, I kind of know Elisabeth's answer, but what would you have done if you hadn't been a scientist?
I would have been either practical doctor or doctor in a hospital and seeing patients. I think this would have been really my way here.
I think you said something different at the beginning. You had a career choice.
Yes, I would have a terrific pianist, but I'm not sure. I doubt it, actually.
Well, you don't know until you try, right?
Yeah, you always try, and yeah.
I mean, I think Rich will agree. You can't be afraid of things in your career, right, Rich?
No, you got to just do everything. I mean, I'm doing this, which is crazy enough.
Different from what you used to do.
Different from what I used to do. I can't believe I'm still doing this.
All right, we need to finish because there's a talk. So let's wrap this up. That's a TWiV from the European Congress of Virology in Dubrovnik. You can find the show notes at microbe.tv/TWiV. If you enjoy these programs, we'd love to have your support. We don't do advertising. We depend on listener donations. You can go to microbe.tv/contribute. Our guest today, Stéphane Blanc from Plants Health Institute of Montpellier. Thank you, Stéphane.
Thank you very much, Vincent. Thank you, Rick. It was great. A great experience. Thanks a lot.
Vanda Juranić Lisnić is from the University of Rijeka, Croatia. Thank you, Vanda.
Yeah, thank you. I'm so happy that you invited me. I'm a big fan, and my husband is even bigger fan of Vince. So
good. You married the right guy then. Elisabeth Puchhammer-Stöckl is from the Medical University of Vienna. Thank you, Elisabeth.
Thank you also for the invitation. Great to be here. Thank you.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. And I want to thank everybody at the meeting. This has been a very hospitable place, and I hope that TWiV will give you a little payback and give you the TWiV bump globally.
I'm Vincent Racaniello. You can find me at microbe.tv. Thanks to Stéphane Blanc and the meeting organizers for having TWiV at ECV 2025, and also Matteo Iannacone for suggesting originally to do a TWiV at this meeting. And I just want to say, European virologists carry on, because as I said earlier, the future of virology in the U.S. is uncertain. It's all up to you, of course, in other countries as well. And from what we've heard today, it's in great shape. Thanks so much. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.